Hi class, in this lecture we are going to be talking about mutations. Now let us talk about mutations. Mutation is the change in the phenotype due to the change of DNA sequence, which affects the mRNA and then affects the primary and secondary structure of the protein. So if it affects the DNA sequence, this can have huge roles on how the protein looks. So when that happens, we call it mutation. If it is a normal or not affected gene, we call that wild type. If it has some mutations in it and it does some other phenotype, we call this a mutant gene. So typically we talk between a wild type and the mutant gene. If the mutation leads to a mutant gene, it shows a variance in morphology, nutritional characteristic, genetic control, resistance to chemicals, and many other things that could happen. Let us move on and talk about some mutations that can occur in DNA. One of the ways we can isolate a mutant is through replica plating. Suppose we wanted to use replica plating to identify a bacterial cell that had lost the ability to synthesize the amino acid histone. First, about 100 bacterial cells are inoculated onto an agar plate. This plate, called the master plate, contains the medium that histone on which all cells will grow. After 18 to 24 hours of incubation, each cell reproduces to form a colony. Then a pad of sterile material is pressed over the master plate, and some of the cells from each colony adhere to the pad. Next, the velvet is repressed down on two or more sterile plates. One plate contains a medium without histone, and one contains a medium with histone on which the original or non-mutant bacteria can grow. Any colonies that grows on the medium with histone on the master plate, but not that can synthesize histone, will not be able to grow on the medium without histone. The mutant colony can then be identified on the master plate. Replica plating is a very effective means of isolating mutants that requires one or more new growth factors. Now, let us talk about how we can do mutations. There are causes of mutations. Spontaneous mutations happen naturally in the DNA due to errors in replication. This was the example I just showed you with the replica plating. You did not do anything to the DNA of the bacteria. You were just testing colonies and looking for the ones that did not grow in a specific type of media. If you have the mutants, they are natural mutants. However, most of the time, when we are talking about science, we induce mutations. We either change the DNA by manipulating the bases or expose it to different chemicals, radiation, or other things to change the DNA base physically. They are known as induced mutations. So, the induced mutations are the ones that are manipulated by our hands, whereas spontaneous ones are the ones that occur in nature. Here is the list of different chemicals on the slide that change things. Again, some of these things are pretty nasty. Radiation, obviously, can also play a huge role in changing the DNA as well. This can affect the DNA and produce mutations in DNA. So, you physically change or manipulate the DNA. We call that induced mutations. However, when it occurs in nature, that is called spontaneous mutation. Alright, here is the list of the different types of mutations that occur. Point mutation means addition, deletion, or substitution of new bases in the DNA, including missense mutation, nonsense mutation, silent mutation, back mutation, and frame shift mutation. Again, point mutation means that you have changed some base pairs in the DNA. However, this can affect a lot of different things. Missense mutation causes a change in a single amino acid. For example, let's see you had AGG, and you now change it to AAG. That is going to change the amino acid. That is called missense mutation. The nonsense mutation changes a normal codon into a stop codon. That is bad because it stops the protein where it is, especially if it's in the beginning or middle of the protein. So it might make the whole thing falls apart. This is what happens to many people when they are missing proteins. Like people that have hemophilia or color blindness, they do not make a complete protein based on their genetic mutation. The silent mutation alters the base but does not change the amino acid. It means it would change the three-letter words. However, it does not change the amino acid. We call that a silent mutation because it does not change the phenotype. An example of this could be you change the three-letter words from AAA to AAC. Since they code the same amino acid, you did not change the amino acid even though you changed the DNA. We also have examples called back mutations, where base composition, when a mutated gene reverts to its original wild-type base composition. So you have a mutation and you change the DNA again, and now it goes back to the original protein. That is called back mutation. You can revert to the original DNA sequence. Another thing that can occur is called a frame shift, and this is basically when you add or delete a base, resulting in the alteration of the mRNA's reading frame. Let us see, if you have ATG and you add another T, and you now have ATTG, so you have shifted everything over. It not only affects the first amino acid, but it affects everything that downstream of that amino acid. Mutations happen all the time. Since mutations can be potentially fatal, the cell has several enzymatic repair mechanisms in place to find and repair damaged DNA. We get exposed to chemicals, things like that all the time. But we are not dying of cancer every day because we have lots of different repair mechanisms out there to fix these mutations. First, when the DNA polymerase proofreads nucleotides during DNA replication. If it make a mistake during DNA replication, it go back and fix it. The DNA polymerase allows it to go back and fix the mistake. There is also mismatch repair. So another enzyme locates and repairs mismatched nutrition bases that were not repaired by DNA polymerase. Light repairs for UV damage. This is really helpful, especially if you like to go tanning and do all those different things getting exposed to the sun. And so obviously, we have these enzymes to help fix our skin, but over time, these things do not work and can lead to mutation, such as cancer. So that is why we're emphasizing that do not use tanning beds, because the light repair only does so much. Then, excision repair locates and repairs the incorrect sequence by removing a segment of DNA and then add the correct nucleotides. So there are lots of mechanisms out there that repair DNA. There is always mistakes happening while we are doing replication. So these are the checks and the balances. Just like anything else, you need to have the checks so you can fix the mistakes that are happening all the time. Here is the proofreading. I will show you this video, which shows you how to go along. This is DNA polymerase fixing the DNA.
Although mistakes can happen during DNA replication, they are extraordinarily rare. A key reason for this is the proofreading function of DNA polymerase. Roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will be a mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase III.
Okay, we talked about proofreading. It is just nice to see how some of these mechanism work. This is an example of how DNA polymerase works. Another thing is the Ames test. About 90% of the substances found by the Ames test to be mutagenic have also been shown to be carcinogenic in animals. The Ames test is essentially testing to see if the chemical can mutate DNA. So what goes on here is you have a mutant strain of Salmonella typhimurium that has lost the ability to synthesize histone. Then you put it on this chemical, and if the bacteria can now utilize this histone, that's bad, because basically it now mutated all these bacteria to use histone. Agricultural, industrial, and medicinal components are screened using the Ames test. So again, the Ames test is based on the observation that exposure to mutant bacteria to mutagenic substances may cause new mutations that reverse the effect of the original mutation. Mutations leading to non-functional proteins are harmful, possibly fatal. We have already talked about negative effect. Sometimes mutations can be helpful, and so they are maintained in the population. What happens is it allows the bacteria to grow better and get into new environments. So we sometimes term these positive mutations. However, again, organisms with mutations beneficial in their environment can readily adapt, survive, and reproduce. So these mutations are the basis change in the population. The population will retain any change that confer advantage during selection pressure. So not all mutations are a bad thing. It can be positive as well. Okay, let us see if you know your mutations. Which of the following mutation could cause a frame shift mutation? Is that a silent mutation, missense mutation, nonsense mutation, or deletion mutation? I'll give you a second. If you said deletion mutation, you are correct. Good job, you got it.